Good morning and good afternoon to all of you for joining our live webinar today. Channelopathies resolved via cryo-EM and X-ray crystallography, an investigation of muscle excitation-contraction coupling. My name is Jason Villagomez, Marketing Manager at Nanion Technologies, and will be your moderator for today's event. Joining us as presenters will be Dr. Conrad Weichbrodt, Product Manager of the Orbit product family at Nanion Technologies, and Dr. Filip van Petegem, Professor Biochemistry and Molecular Biology, University of British Columbia. Today's presentation is as follows. Conrad will provide an introduction to Orbit technology platforms used for experiments on lipid bilayers and covering the basics and principles behind the Orbit technology. Filip will then proceed and present on his work regarding channelopathies resolved via cryo-EM and X-ray crystallography, muscle excitation-contraction coupling. Upon completion of the presentation, we will hold a live Q&A session, and as such, welcome you to ask our presenters questions throughout the course of the presentation. You can log a question via the chat window found on the right-hand side of your screen. And if you happen to experience any technical difficulties at any time, please inform us via the chat box. Additionally, today's presentation will be made available on demand shortly upon completion of the event, inclusive of a transcribed copy of the Q&A. If you have any additional follow-up questions, please do not hesitate to get in touch with us and our presenters, and we can forward you the contact details after the presentation over. I will now hand over to Conrad for the start of the presentation.
Thank you very much, Jason, for the nice introduction. So today's webinar will be about channelopathies and especially on Ryanodine receptors. And this will include electrophysiology on these transmembrane proteins. So please let me swiftly introduce the devices that Nanion offers for research on such species. So why would anyone, if you think about electrophysiology on transmembrane proteins, what comes to people's mind is normally whole cell patch clamping, where you would record currents through a whole living cell, and Nanion offers various devices for this. Please drop us an email if you're interested in these devices. Something fundamentally different, however, would be experiments on artificial lipid bilayers. So the whole chip and the recording substrate would be differently. You would generate a bilayer first and then introduce the protein or target of interest. And we offer the devices of the Orbit family for these kind of applications. So why would anyone prefer doing lipid bilayer experiments to whole cell patch clamping? Well, first of all, you could reach target proteins that are in non-patchomal membranes, for example, in the membranes found in cell organelles, like, for example, the Ryanodine receptor from the Sarcoplasmic Reticulum in muscle cells, as we will learn later on. And I'm really looking forward to that talk because recording those is not an easy task, and Filip will show excellent traces from his most recent work on these channels. Also, you can purify your, for example, ion channel of choice and integrate only this ion channel inside a pre-built artificial membrane. So you will not have to deal with backgrounds like in living cells. You will not have to deal with artifacts. And you can also investigate antimicrobial peptides, toxins, DNA pores, or basically anything that will form a pore inside a lipid bilayer. And if you already have a pore, you could send something through and do nanopore analysis, as we will see later on in the example traces I will show. So we have two devices right now for experiments on artificial bilayers. It's the Orbit Mini with 4 recording channels and its bigger brother, the Orbit 16TC, that we recently introduced. So please check out our homepage for more information about this, or just drop me an email. Both are rather small and really easy to use. The Orbit Mini is as big as an external hard drive. The Orbit 16TC is as big as a shoebox. You just connect it to a laptop or a computer. They are their own Faraday cage and you're basically ready to go. And both offer temperature control. Both devices include amplifiers of our cooperation partner Elements. These amplifiers are tailor-made for recording of single channels. They have great noise levels, and you can record the 4 channels of the Orbit Mini or the 16 channels of the Orbit 16TC at sampling rates up to 200 kilohertz per channel. So there's no multiplexing involved. You can record them fully in parallel. And how does the recording substrate actually look like? Well, we use the microelectrode cavity array chips, so-called MECA chips. And on the surface of these chips, you would find cylindrical cavities that you would basically span with a lipid bilayer. I will show on the next slide how this is actually done. And the bottom of these cavities would be the driven electrode for each channel. And they share a common ground electrode, which is the rim of the chip. And of note, the chip is its own recording chamber. So for one target, you could use one chip and never have to deal with cross-contamination. These chips can be used several times for several experiments, and they are very robust and easy to clean. So how do I actually generate a membrane there? Well, it's the classical painting approach, like any BLM you would find on other setups as well. So you could apply lipids in organic solvents over the cavities in this chip. And for the Orbit Mini, we would recommend to use the air bubble approach, meaning that you have your cavity filled with buffer and your chip filled with buffer. And then you would use a pipette tip. And this tip is not filled with lipid solution. It's only dipped inside the lipid solution. So the pipette tip will be covered with lipid solution, an organic solvent. And then you actually form an air bubble over the cavity. So if this air bubble reaches the other side of the cavity, the lipids can actually adhere to the other side of the cavity, and upon sucking the air bubble back in, you will form a bilayer. And I took this figure from a book chapter we recently published. If you are interested in this, and if this might be interesting for you, just drop me an email so I can make this available. And of note, on the Orbit 16 TC, the whole painting process for all 16 membranes at once is automated, as I will show later on. So a frequently asked question would be, can I record with asymmetric buffer solutions? Because I have this cavity, so I cannot really reach the trans side anymore. But yes, I can fill the whole chip with the trans solution for my later experiments. I can then build the membrane, and I can exchange the cis solution after I build my membranes, and the membranes are so stable that they can absolutely survive that. So I would end up with asymmetric conditions. I can do this exchange simply with a pipette, and I can build up pH or calcium gradients and trap activators below my membrane on the trans side, and so on. And both systems have an integrated temperature control. So the temperature control would be connected to the device. You do not need to use it, but it will enable you to apply for the whole recording's temperature in a range between 5 and 50 degrees Celsius. So you can actively cool or heat. It's really simple to use. It's integrated in the software. You just set the temperature you want, and several sensors inside the system will hold this temperature constant for virtually for hours. And this is really beneficial because you can work at physiological temperatures, and you can work on species that are heat or cool activated, like trip channels. For example, the cold activated trip A1, you can activate it by lowering the temperature, or the heat activated trip V1, you can activate it by actually rising the temperature. Or you could alter kinetics of a certain experiment. So this is a proof of concept experiment we frequently use. It's translocation through the alpha hemolysin pore. And I really like this as a proof of concept experiment because alpha hemolysin is commercially available. Anyone can buy it. It's not even that expensive. So you could try for yourself, and you can send molecules through and then see transient blockages in the current signal. And by cooling everything down, you could make these events last a bit longer, so you could catch events you would otherwise miss. And we did this on the Orbit Mini, and we could really nicely see that we have an increase in open pore current, as we would expect with rising temperature, and also an increase in the frequency of events as more molecules would pass through the pore. And for our recently introduced Orbit 16 TC, how does that look like? In the inside, it has two covers, an outer one, just shielding the whole experiment from the environment, and then you have an inner lid, a smaller black inner lid. This is the Faraday cage. So if you apply a chip and your steer bar, that will paint the membranes, and you apply the lower lid, you can still reach the chips while recording at highest precision. And in the MECA 16 chips, you would actually have 16 of these cavities that are all spanned by individual bilayers just at the push of a button. And we did the same proof of concept for this device as well. So this is how the whole setup would look like. You have the Orbit 16 TC device, you have the temp control and a laptop. So it's really easy to just put it on a different bench. You could basically work in a train if you want. And also here we did our proof of concept experiment, and we also could generate really nice data, seeing that we have an increase in open pore current and event frequency with rising temperature. So please feel free to directly approach me if you if you're interested in our devices. And with this, I would hand over back to Jason. Thank you very much.
Thanks, Conrad, for the quick introduction. I now go ahead and turn over to Filip, who will present his work on channelopathies resolved via cryo-EM and X-ray crystallography, muscle excitation-contraction coupling. So Filip, the floor is yours.
Okay, well, thank you, Jason and Nanion in general for putting this together and allowing to present my work. So yeah, thanks for signing in this morning or this evening, wherever you are. And today I'd like to talk to you about channelopathies and particularly the use of structural biology to gather some insights. And I will start straight away with a particular channelopathy that's shown right here. What you're looking at here is an electrocardiogram from a patient who is at risk of sudden cardiac death. And so the patient has a genetic condition. It's called CPVT, or catecholaminergic polymorphic ventricular tachycardia. So that's a mouthful. But essentially, if you measure an electrocardiogram from such a patient under resting conditions, it actually looks quite normal. It's only when you ask the patient to do some mild exercise that you can see, at least under these conditions, the ECG is no longer normal, and the patient is at risk for developing ventricular fibrillation, which can result in sudden cardiac death. So this is a very serious disorder. It's stress-induced, and it's a genetic condition due to mutation in this protein, the RYR2. Now, a very related gene or related protein is RYR1. RYR1 has a completely different type of disease associated with it, and the most famous one is malignant hyperthermia. Now, malignant hyperthermia is a kind of a condition that arises with a combination of a mutation, typically in the RYR1 gene, together with a triggering agent. And a triggering agent could be a volatile anesthetic, such as the ones used during surgery. And it's really that combination of these two factors that can lead to a massive rise in body temperature, muscle rigidity, and a whole range of downstream effects that can actually cause you to die during the surgery. This is one of the diseases associated with the RYR1 gene. There are a couple of others, most notably central core disease. It's a type of progressive myopathy, so very severe muscle weakness. So all these disorders, the inherited cardiac arrhythmias, and here these skeletal muscle disorders, they're very different, but at the biochemical level, they really have a lot in common. So both of these genes, RYR1 and RYR2, then code a protein known as the Ryanodine receptor. And so if you're not familiar with this, it's this big ion channel that's located here on the bottom of my slide. You typically will find it in either in the endoplasmic reticulum or the sarcoplasmic reticulum membrane. And its primary role is to release calcium. So for example, in a cardiac myocyte, let's assume that on the top here, you're looking at the plasma membrane. At the bottom is the SR membrane. And at very specialized sites, these two membranes are in close proximity, and that allows functional crosstalk between various ion channels. So what will happen is that if you have a depolarization of the plasma membrane, is that this can trigger opening of this L-type voltage-gated calcium channel. As a result, calcium will flow into the cytosol. That can bind to a nearby Ryanodine receptor, which is then triggered to open, and which allows release of calcium stored from the SR also into the cytosol. And so the primary role of the Ryanodine receptor here is the role of a signal amplifier. Essentially, it detects some calcium and it amplifies that calcium signal. Now, in the human genome, there are 3 different isoforms that are encoded: RYR1, RYR2, RYR3. RYR1 is the one that you primarily find in skeletal muscle. RYR2 is the main isoform you find in the heart. RYR3 was originally found in the brain, but it's expressed in multiple cell types. And in fact, all 3 isoforms are expressed in many different cell types. It just happens to be that they've mostly been studied in the context of muscle tissue. And so, as I told you, Ryanodine receptors are targets for disease mutations. And most of these mutations, not all, but the bulk of them, are what we call gain of function disease mutations. So what that means is that the Ryanodine receptor can open under conditions where that shouldn't happen. That's a gain of function. And now calcium can leak into the cytosol and do a lot of really bad stuff. And among the things that could happen, I told you about these genetic diseases: malignant hyperthermia or central core disease. That's due to mutations in the skeletal muscle isoform. The inherited arrhythmia, the stress-induced arrhythmias, due to mutations in the cardiac isoform, RYR2. But there's also emerging evidence that Ryanodine receptors are involved in maybe non-genetic diseases, but that misregulation of the Ryanodine receptor can either cause or exacerbate a number of disorders. And that includes atrial fibrillation, cardiac arrhythmias that arise during heart failure. And there's also emerging evidence that misregulation of the Ryanodine receptor plays a big role in the progression of Alzheimer's disease. So as a result, it's of course of interest to try and understand how does this misregulation of the Ryanodine receptor exactly occur, both at the genetic level, but maybe also at the regulatory level. So I'm primarily a structural biologist, so I like to look at structures. Now, the Ryanodine receptor is a huge protein, 2.2 megadalton in size, and that makes it a key target for cryo-EM studies. And in fact, many studies have been performed already in the past. Already since the 1980s, there was a negative stain EM imaging that was done. I'm showing here a study. This is a particular structure from Irina Serisheva's group, which just highlights the overall architecture. And so it resembles the shape of a mushroom. The stalk of the mushroom is essentially what's crossing the membrane of the ER or the SR, and the cap of the mushroom is located in the cytosol. And so what you can see from the top view, if I allow it to rotate again, is that this is clearly a 4-fold symmetrical protein. So it's encoded by 4 identical subunits of around 550 kilodalton each. And this actually makes it at least the largest known ion channel that we currently know. Now, at that time, before major developments in cryo-EM, we're actually trying to peer into the details of this protein by solving crystal structures of various bits and pieces. And so this here shows an overview of various crystal structures available that are shown by different colors. And we could map these, we could dock these into the cryo-EM maps. But of course, technology has changed a lot, and there is now a very good cryo-EM available. And so essentially since 2015, early to 2015, a couple of high resolution cryo-EM structures were published back to back, and allows us to really look at RyRs in a lot more detail. So the transmembrane region here is very well resolved. You can see all the alpha helices. In general, you can already see by looking at these cylindrical densities that the Ryanodine receptor is rich in alpha helices in general. Now, this is a big protein, and if I want to talk to you about just the structure, I could fill an entire talk just talking about all the details, and it's a bit of a daunting task to do as well. So I just want to simplify the architecture a bit. And so if we look at just the business end of the protein, which is the transmembrane region, and I call it the business end because this is the part where calcium ions can permeate the membrane. So what I'm showing here is a cartoon form of the transmembrane region. And for clarity, I'm only showing 2 out of the 4 subunits. And what you will see is that in the center, there's a so-called pore-forming domain. And that's a module that you find in many different ion channels of the tetrameric ion channel family. Adjacent to these pore-forming domains are these 4 helical bundles that resemble voltage sensing domains that are found in voltage-gated ion channels. And so essentially, Ryanodine receptors are clearly related to voltage-gated ion channels that don't have proper voltage sensing, but evolutionarily, it's clearly that they're related. Now, this is the business end of the protein. Built on top of that is a huge core of so-called alpha solenoids. And these are just very alpha helical rich motifs. There's a couple of these very large solenoids that I'm showing in different colors here. And these alpha solenoids constitute around 50% of the mass of the entire ryanodine receptor. And then on top of these alpha solenoids are a number of globular domains, which I've named here and circled. These are domains that are primarily either rich in beta strands or form these horseshoe-shaped structures. They have evolved to have different functions. We don't know all of the functions yet, but some of them would receive binding of ligands, others would receive post-translational modifications. And if you really think about it, this whole cytosolic shell here, this cap, its primary role is really to dictate what is happening here in the transmembrane region. And so as you can imagine, if you have disease mutations pretty much anywhere in this protein, this can affect the function of the channel by interfering with opening and closing of the pore. Okay, so this is just the general architecture, really just very brief. But in recent years, what we've mostly been doing is study this ryanodine receptor and its regulation at various levels. So we've been taking a lot of looks at disease mutations. Also, we've been looking at how post-translational modifications are occurring, particularly how kinases can act on the ryanodine receptor. We've also used cryo-EM and are using cryo-EM to look at the binding of various small molecules and see how they affect the gating behavior. And also a very interesting question, which is how ryanodine receptors couple mechanically to voltage-gated calcium channels in skeletal muscle. Now, in the interest of time, I'm actually only going to spend some time here on these top two topics, particularly disease-causing mutations in the ryanodine receptor and the regulation by kinases. And so I told you before that in the past, we were able to solve many different crystal structures of bits and pieces, which allowed us to map disease mutations with pretty high precision. We could analyze their chemical environment quite well. And we could also easily compare structures of wild types and disease mutations because these crystal structures were obtained from parts of the ryanodine receptor that are expressed recombinantly. So it was actually very easy to introduce your favorite point mutation and see what the effect is. And so I'm actually not going to show these data, but it's been published before. And the bottom line is that we found that many disease mutations are actually clustering at the interfaces between these different color domains. And when we actually compared structures, we saw that in many cases, the disease mutations were triggering conformational changes in the isolated pieces that we crystallized. Now, of course, there's a limitation to this study. It's because we're only looking at fragments of the ryanodine receptor. And so if you really want to understand the complex effect of a disease mutation, it's important to look at it in the context of the full protein. And so the question is, how can we get a cryo-EM structure of a disease mutant form? And so, of course, you could just say, well, just introduce your favorite mutation and purify that. The problem is that the ryanodine receptor is a pretty large protein. And even though you can produce it recombinantly, it's a bit painful to do so. And most cryo-EM studies to date have been performed on ryanodine receptors isolated from native material. So either from skeletal muscle, for example, rabbit skeletal muscle, or from pig hearts. And so you just use the native protein that's available. And it's very straightforward to do. The only problem is, in that case, you're stuck with the wild type protein. So how are you going to compare with the structure of a disease mutant form? So what we did, we actually took the path of least resistance, and we actually turned our attention to pigs. Because there are pigs with a certain syndrome called porcine stress syndrome. Now, porcine stress syndrome is a kind of disorder that's very similar to malignant hyperthermia in humans. It is also differs a bit because it can also be induced by stressful conditions. And it was found actually by the lab of David McLennan that the causative mutation for porcine stress syndrome is a mutation in RYR1, the skeletal muscle ryanodine receptor, and it has this arginine 615 to cysteine mutation. Now, the phenotype here is quite strong. If these pigs get stressed, they essentially can die. So you could ask the question, how is that evolutionarily even possible? How can such a pig survive? And you could argue that there's actually one selective advantage for having this mutation, if you can call an advantage. That is the fact that the meat tends to be leaner. So these muscles, these pigs actually have a lot less fat content. So it's kind of a way to produce leaner pork meat. We actually got in touch with a farmer in Iowa who was still keeping these types of pigs. So we ordered a few kilograms of meat that essentially allowed us now to purify ryanodine receptor containing this disease mutant. Okay, so as Conrad nicely introduced, we also did some bilayer electrophysiology to characterize this mutant and verify what it really is doing, particularly under the conditions that we also want to investigate via cryo-EM. And so on the left here, you see single channel traces of wild type big ryanodine receptor. And we're looking at recordings performed at different calcium levels. And you see at very low calcium levels, there are very few openings. These openings increase with increased calcium concentrations and then decrease again as you go up to the millimolar range. Now the precise numbers don't matter here. What really matters is when you compare wild type and disease mutant. So on the right, you see the similar conditions used for the R615C, so that disease mutant big RR1. You can see that at every calcium concentration tested, clearly the open probability is substantially higher. And so you can also plot this, and you can see here the trace for the wild type in black, and the mutant clearly has increased opening. So this is a typical gain of function mutation, which is entirely what was described before and expected for either malignant hyperthermia or porcine stress syndrome. Okay, so that's the characterization initially. Now, how about structure? So we first had to have a reference structure of the wild type big RR1, and that's shown here on the left. This is the cryo-EM map of wild type big RR1, and this is the corresponding model here on the right that's been built into that map. And so I've colored this structure according to different regions, and I'm actually going to point out why that's important to look at these different regions. So overall, this structure is similar to what was described before for rabbit RR1. They're very related. But let me point out exactly where the disease mutation is. And it's actually indicated here in a black stick, which you can't see, so I'm highlighting it here. There's a little black residue here that sits at the interface between this blue solenoid region and this green solenoid region. And it's actually just that very subtle mutation that can actually wreak a lot of havoc. So let's zoom in on that. Okay, so showing here again is the cryo-EM map of the wild type big RR1. And so this arginine 615, its side chain is actually very well resolved in the cryo-EM density. And you can see it's actually strategically located at the interface with two other domains. Okay, so it's part of this blue solenoid region, and it forms interactions with this green solenoid and this beige solenoid. So it really sits at the domain-domain interface, and this is actually where you typically find disease-causing mutations in the ryanodine receptor. Okay, so what is it actually doing? So you might imagine that mutating this to a cysteine is clearly going to affect the way that these domains interact with one another, but could it also trigger a conformational change? So what I'm going to show now is an animation that toggles between the structure of the wild type, which is what you're looking at now, and the disease mutant. So we got this blue and this green solenoid regions for those in the field. So these are called the N-terminal solenoid, and the green is the bridging solenoid. That's just a nomenclature. And arginine 615 is here seen at this interface. And so if this animation plays well, we're going to toggle between wild type and disease mutant. And what you hopefully can see is that introducing this mutation triggers some pretty large conformational changes. And you can describe it as kind of a tilting motion. So relative to this blue solenoid, you see this green solenoid and other parts of the ryanodine receptor actually tilting away. And so the tilt is, of course, small, relatively close to the hinge point, but the further away you go from that hinge point, these movements are actually large and they're in the order of around 10 angstrom. Okay, so just interfering with this interaction between these domains here, this can cause a large tilting motion, a pretty large conformational change. Okay, so I'm going to zoom out and now look at the overall conformation of the Ryanodine receptor in wild type and disease mutant form. So again, let's animate this. And what you will see once you go to the mutant is that there are lots of motions throughout the Ryanodine receptor. You may appreciate here in the center that these different pieces are pulling apart. And actually, if we zoom in here to the transmembrane region, you see that the pore forming region, which is shown in red, is actually getting wider. And in fact, the disease mutant is in an open state. So the mutation under these conditions used triggers opening of the Ryanodine receptor. And here's shown a side view where you can also see substantial conformational changes. Okay, so here we have a disease mutant that clearly affects the structure of the Ryanodine receptor, and under these conditions actually triggers an open state. And so let me simplify this. For the wild type, we have a structure of the Ryanodine receptor in a closed pore. There is a certain angle between the N-terminal solenoid and the bridging solenoid that arginine 615 is right at that hinge point, kind of keeps them together. If you mutate that to a cysteine under these conditions, we got this large change in the tilt angle, and this results in an open pore. Okay, so obviously wild type, disease mutant, it's a gain of function. It's kind of expected that opening is facilitated, but of course under physiological conditions, this mutant does not always display an open structure. Obviously it's still a channel that gate. So if we want to get a full picture of what's going on, we really would want to compare all the different states. And minimally, we'd like to compare a closed wild type structure with a closed disease mutant and an open wild type and an open disease mutant. So right now we just have closed wild type and open disease mutant. So to do that, we actually turned our attention to also some auxiliary proteins. And so in particularly what we wanted to investigate is the effect of camodulin. So camodulin is a very famous protein, binds calcium. And under apo conditions, under calcium-free conditions, it was previously reported that this can increase the open probability of the skeletal muscle Ryanodine receptor. And this is something we also see. So under these conditions used here, under essentially normally calcium-free conditions, you see that for the wild type, when you add apocamodulin, you get an increase in open probability. But oddly enough, that's not the case for the disease mutant. In fact, with the disease mutant, if anything, you might actually have a decrease in open probability, although that's not statistically significant. So anyway, we wanted to investigate this structurally and also hoping to capture some additional conformational states of the Ryanodine receptor. And this is actually what we were able to do. So shown here is a cryo-EM structure of that pig RYR1. We have different domains colored in, differently. But camodulin, apocamodulin, is this purple protein here that sits at the periphery of this cytosolic cap. And on the right here is kind of a blow up of this, and you can see it interact actually mostly with this green solenoid, this so-called bridging solenoid. Now I can tell you a lot about the precise interactions with camodulin, but what I want to highlight here is actually what it allowed us to do in terms of analyzing the disease mutant. So there's a certain pipeline that you can follow when processing a cryo-EM data set. And this may look complicated, but essentially what it allowed us to do, we had data sets for the wild type RYR1 with apocam, also for the disease mutant RYR1 with apocam. And essentially for each, we actually got two different structures. And so for the wild type, we actually got 70% of the channels appear to be in a closed structure, and about 30% were open. Whereas for the disease mutant, it's almost inverted. You've got a smaller proportion of closed channels, but most channels are actually open. So again, this really nicely shows this gain of function effect, but now on the structure. You go from a certain distribution of closed and open channels towards more open channels for the disease mutant. But now, of course, it allows us to do these pairwise comparisons, look at closed structures versus open structures. Okay, so let's have a quick look. So again, I'm zooming in here on this area that contains the disease mutation. So this is arginine 615, forms this hinge point between the blue and the green solenoid. And so we're going to toggle now between structures of wild type and mutant, but in this case, we're only comparing closed structures. Okay, what hopefully you can see, maybe with some delay in this animation, is that there again is a tilting motion visible. Also, when you look at the overall structure, you get overall motions here that are very similar to the motions I showed you before, but they're a little bit dampened. And most important here, there are very few changes in the transmembrane region. You might argue that the pore gets slightly wider, but it's still not wide enough to actually permeate calcium ions. And also here on the side of the cytosolic cap, you see some motions that we typically see before. So essentially what's happening is that even in the closed channel, you kind of see that this mutant goes into a conformation towards channel opening, but it's not as extreme yet as with an open channel. It's like a partial movement towards opening. Okay, now let's look at the open state. So let's toggle between structures of an open wild type and an open mutant. And I'm just going to show here the top view. And bottom line is that if you have a hard time figuring out what the motions are, that's okay, because the motions here are very subtle. And in fact, any tiny thing that you see here might actually just be explained by differences in resolution. So in the open states, there is actually very little difference between wild type and mutant. It seems that the mutation really affects the closed state, but also obviously the ability of shifting between closed and open. Okay, so let me summarize this a little bit better. Let's simplify it. So essentially for a wild type RR1, there are of course closed state structures. In these closed states, there is a certain tilt angle between the N-terminal and the N-terminal solenoid and the bridging solenoid. Arginine 615 sits at that hinge point. For the channel to open, you need to have a change in this tilt angle. And essentially, you can imagine that this is going to cost some energy, because this arginine is involved in interactions with this bridging solenoid, altering these interactions. So normally, channel opening would only occur if you can deliver some energy to the system. So you need some activating ligand to bind, such as, for example, calcium. But if you now have this hinge point broken, if you introduce a cysteine in here, two things happen. One thing that happens is that there already is a change in the tilt angle between these two solenoid regions. It's actually already increased. For a channel to open, it needs to increase more, but this seems to be facilitated because now there's less energy required to break the interactions mediated by residue 615. So we think this nicely explains this kind of gain of function that you can see by functional experiments. And it really seems that this arginine 615 sits at what we would call a very critical position, a so-called labile interface that's involved in channel gating. Now, there are many more disease mutations in the ryanodine receptor, and I think many of those have their individual stories, and there will be a lot to be explored via cryo-EM. But I also pointed out that post-translational modifications also play a role in disease. And so what I briefly want to talk to you about is what we figured out about kinase regulation of cardiac ryanodine receptors. So in the heart, the main isoform is RYR2. And there's been a lot of literature by many different groups to show that several kinases can act on this ryanodine receptor. And this includes protein kinase A, or PKA, and chemogene-dependent kinase 2. Now, these are two very famous kinases. There's a lot of controversy around their role and which one is more important and which sites are being phosphorylated. But a general consensus would be, I would say most people would agree, that these kinases can confer a gain of function to the ryanodine receptor. So if that ryanodine receptor gets phosphorylated, it would be of hyperphosphorylation kinase is involved in the fibrillation.